We completed probe hybridization. Now we will pool and wash the samples.
We have concentrated post-Hyb buffer, thaw at room temperature, and keep on ice. We have enhancer, thaw for 10 minutes at 65 degrees Celsius at max speed on a thermomixer. Vortex, and centrifuge briefly. Keep the enhancer warm. Verify there is no precipitate. Do not keep the thawed enhancer on ice, or the solution will precipitate. We have nuclease-free water, and we have a filter.
Next, we will prepare the post-Hyb wash buffer. Add the reagents in the order that they are listed in the user guide.
Add nuclease-free water, concentrated post-Hyb buffer, and enhancer. Vortex briefly, and keep at room temperature. Do not keep at 4 degrees Celsius. Remove tubes from the thermal cycler after overnight incubation. Dilute each sample by adding 175 microliters post-Hyb wash buffer and pipette mix 5 times. Take 10 microliters of diluted sample and determine the cell concentration by using a Countess automated cell counter, a Celica counter, or a hemocytometer. Calculate the total cell number present in the tube. See tips and best practices for sample counting in the user guide. Accurate cell counting is critical for optimal assay performance. We recommend using a fluorescent dye and automated cell counters.
Now we will pool the samples.
Pool an equal number of cells from different hybridization reactions into a 5 milliliter centrifuge tube. See post-hybridization pooling calculation in the user guide for guidance on calculating the volume of each sample to be added.
Next, we will wash the samples.
Add 2.3 milliliters of post-Hyb wash buffer. Mix by inverting 5 times. Centrifuge each tube containing pooled samples at 850 RCF for 5 minutes at room temperature. Remove the supernatant without disturbing the pellet. Resuspend the cell pellet in 1 milliliter post-Hyb wash buffer and transfer to a 1.5 milliliter microcentrifuge tube. Incubate at 42 degrees Celsius for 10 minutes in a thermomixer. Centrifuge at 850 RCF for 5 minutes at room temperature. Remove the supernatant without disturbing the pellet. Resuspend the cell pellet in 0.5 milliliter room temperature post-Hyb wash buffer. Pipette mix 5 times. Incubate and perform the wash again. Incubate the sample at 42 degrees Celsius for 10 minutes in a thermomixer.
Next, we will prepare the post-Hyb resuspension buffer. Add the reagents in the order that they are listed in the user guide.
Add nuclease-free water, and concentrated post-Hyb wash buffer. Pipette mix 10 times and maintain at 4 degrees Celsius. Remove the samples from the thermomixer and centrifuge at 850 RCF for 5 minutes at room temperature. Remove the supernatant without disturbing the pellet. Resuspend the cell pellet in an appropriate volume of chilled post-Hyb resuspension buffer. The volume of the buffer will depend on the starting number of cells in the pool. In this case, we had 7 million starting total cell number in the pool. Therefore, we will use 1,000 microliters of post-Hyb resuspension buffer. Pipette mix 20 times to resuspend and break up any cell clumps, and maintain on ice.
Finally, we will filter the sample.
Filtration is essential for best microfluidic performance. Pass the sample through a 30-micron filter into a new tube. Lightly touch the filter membrane with a pipette tip at a 45-degree angle where the filter meets the wall, and pipette directly through the filter. Tap or spin down if liquid remains in the end of the filter. To maximize recovery, any residual volume can be pipetted from underneath the filter. If the sample does not go through the filter, pipette the sample back into the pipette tip and reapply to the filter. Determine the cell concentration of the sample using a Countess automated cell counter, a Celica counter, or a hemocytometer. If the sample concentration is not sufficient to achieve the desired target cell load, refer to the user guide for additional instructions. Proceed immediately to the next step.
We have filtered and counted the samples. Next, we will prepare the gem master mix and dilute the samples.